everyone, welcome to my presentation. I'm very excited to talk to you about our latest review, the curating and comparing of 114 genome-scale metabolic models of Staphylococcus aureus. Staphylococcus aureus is one of the most prominent and modern pathogens we have today, and approximately 1/3 of us is colonized with Staphylococcus aureus in our noses, usually asymptomatically. However, in some cases, the disease starts to spread. Staphylococcus aureus infects us, and this causes high morbidity and mortality worldwide. The main problem about Staphylococcus aureus are its multi-resistances, and we have to find some new ways to fight Staphylococcus aureus. To do so, we can use genome-scale metabolic models. With genome-scale metabolic models, we can simulate the growth behavior in different environments, but also in interaction with other pathogens or other microbes. Before we can simulate the Staphylococcus aureus in different environments, we need to have a genome-scale metabolic model. And to find out which genome-scale metabolic models are currently available of Staphylococcus aureus, we did some literature research and identified 114 models. We downloaded them and evaluated all of them with respect to their scope, their availability, their format, their growth behavior, similarities, and also their predictive value. To start with, we want to look at the properties of all available Staphylococcus aureus models. We found 7 publications about Staphylococcus aureus and genome-scale metabolic models. The first was published in 2005 by Becker and his colleagues, and they manually curated a genome-scale metabolic model of Staphylococcus aureus. Same accounts for the model of Heinemann and his colleagues, which was also published in 2005 and also manually curated. And the latest publication of a Staphylococcus aureus model from 2019 was published by Seif and her colleagues, and it was also manually curated. From the Path to Models project, they automatically curated a collection of 33 Staphylococcus aureus models, different strains, and the largest collection of different strains was published by Bolsi and his colleagues in 2016, and they published 64 different models. We also evaluated the models regarding their MeMode score, which is a testing suite, and the higher the MeMode score, the more annotations are available, and also the better connected is the model. We also tested the initial growth conditions or the initial growth behavior, whether the model is directly simulatable upon downloading, and this was the case for all of the models except for the collection from Lee et al. And the Path to Models project. You can also see that the models are mostly available as SBML file formats. However, the model by Lee et al. Or the models by Lee et al. Were only available as Excel spreadsheets, and we tried to convert them into SBML models, which can then be simulated. Simulating also means simulating the growth in different media, in different environments. We know that Staphylococcus aureus can grow in the human nose, so the synthetic nasal medium SNM should be simulatable in all of the different Staphylococcus aureus models, but we also know that Staphylococcus aureus colonizes the lungs of patients with cystic fibrosis, so we tested the synthetic cystic fibrosis medium SCFM. It is also known that Staphylococcus aureus can grow in the gut, so we have a gut medium, and a chemically defined medium which can also be supplemented with glucose and galactose. And interestingly, the model by Heinemann et al. Was the model that grew on all the different media which we tested in our analysis. Finally, we also wanted to compare the models with respect to their gene set. When we're looking at genome-scale metabolic models, the models are based on the genome and the genes encoded in this genome. So when we're looking at one specific strain, for example the USA 300 FPR3757, which is a multi-drug resistant strain of Staphylococcus aureus, in general, all the models should have the same gene content because they're all based on the same genome. However, when we compare those models, we see that only 15.7% is shared of the gene set among all the different models. This number is a bit higher for the strain N315, which is a methicillin-resistant strain of Staphylococcus aureus, which is sharing approximately 40% of its genome or the gene content across all the models. So it does not only account the genes, but also probably the curation and reconstruction method makes a huge impact on the gene and the model content in the end. If you're interested further in this study, I will refer you now to our publication in NPJ Systems Biology and Application with the title Curating and Comparing 114 Strain-Specific Models of Staphylococcus aureus. Thank you very much for listening.